Thank you very much for the invitation. So this is the plan. I'm going to go over a little bit the theory behind the history of natural sciences and deep learning. A few words on theoretical aspects of deep learning, and then I'll show you some applications in physics, chemistry, and biology. So natural sciences, you are very familiar with this kind of images, but this is the beginning of natural sciences shortly after the Renaissance, with people like Galileo, who built one of the first telescopes with a magnification factor of 20x. At the time, there were, of course, no computers. All the data came straight through his eye and through his brain. He had no tools really for storing data long-term and analyzing data using proper statistical or machine learning methods. And you see the same things across all the natural sciences. This is for biology, the invention of the microscopes in Holland, one of the first microscopes with a magnification factor of 100x, etc., etc. Even the first stock market, for instance, was created around that time. And today things have changed. We have much larger instruments. This is the Hubble telescope with a magnification factor of 5,000 that are capable of producing petabytes of data very rapidly. And of course, in this case, the data is to be beamed down to Earth and stored and then analyzed using statistical, machine learning, AI methods, such as deep learning. This is the Large Hadron Collider, where you have millions of detectors that are located around the point of collisions of particles. And this particular instrument can produce a petabyte of data per second, which is a gigantic amount of data. Same thing in biology. You have here this instrument. Actually, I saw it this morning. You have it here. It's a DNA sequencer from Illumina, and you can sequence a human genome in an afternoon on this instrument, and it produces data that looks like this. This is the genome of the AIDS virus. It has 10,000 letters of DNA that fits on one slide, but your genome looks exactly the same, except that you have 3 billion letters, ACGTs, in your genome. And of course, this is completely incomprehensible to humans. You definitely need computers, not only to store the data, but to try to understand the data. So when did deep learning start? Anybody has a guess? Beginning of deep learning? Origins of deep learning? Deep learning started with shallow learning. And shallow learning, well, I'll skip this. Shallow learning is here. This is Gauss, who invented linear regressions, the equations for linear regressions. Well, everything about machine learning is already contained in this slide. You have data, you're trying to model the data, you come up with a model. In this case, it has only 2 parameters. You want to fit the model to the data. How do you fit a model to the data? Once you have fitted the model to the data, you can use the model for prediction, or you could compare different models, etc., etc. So all the questions of machine learning really are already present in this slide. There are 2 things that are peculiar to this slide, is that for regression, for linear regression, you can find the parameters of the model analytically. You have a mathematical formula that's going to give you the parameters of the model, all right? But that's true only for linear regression. All other nonlinear models in general, you cannot solve the parameters analytically. You're going to have to go through some kind of optimization, gradient descent, etc., to find the parameters. And the other thing that is peculiar to this case is the fact that it's very interpretable, because it's in 2 dimensions, and your eye sees immediately what's going on. Even linear regression in 100 dimensions is somewhat opaque. And if you go to nonlinear regression or nonlinear classification, then of course you're in a high dimensional space, then your eyes are pretty much useless. And this is essentially what deep learning is. It's just linear regression on steroid, where you're dealing with nonlinear objects, nonlinear surfaces, and you're trying to fit those surfaces, right, using these neural networks. And again, an example into the dimension in the upper right corner, where you're trying to fit a nonlinear curve. You could use Gaussian processes, of course, or neural networks or other things, but it's nonlinear. You can still see things because it's in 2 dimensions. But if you want to go to cases where you have 1 billion of data points in 1 million dimensional space, which is what you have with images, for instance, then of course things are not become very opaque. You cannot really see what the model is doing very easily. So today these deep neural networks have been used for all kinds of problems, in particular for image analysis. And this is just to give you a sense of the breakthroughs over the last few years. This is the ImageNet. It's a benchmark in computer vision. Up to 2010, the error rate was around 30%. This is a network that has to recognize a number of objects. And you can see that using neural networks in the last few years, the error rate has been dropping. And humans on this particular task have an error rate of 5%. And now, using deep learning, the error rate is below 5%. So you see superhuman performance in image recognition. And of course, you have seen in the news now systems that are using deep learning for playing Go, for instance, AlphaGo has a superhuman performance, et cetera, et cetera. So in specific areas, more and more, you're going to see these deep learning systems that are capable of outperforming even human experts. So how did we get into deep learning, and what is it exactly? Well, it goes back to the 1950s, and really there is a very strong inspiration from the neurosciences behind deep learning and neural networks, of course. So in this slide you have in the upper left, this is just to summarize what people knew in 1950 about the brain. And most of the basic concepts were already in place. You have Darwin here. The brain is a product of evolution, so it's a product of a tinkering process. It's a very messy machine. They knew that it is modular. There are areas that are specialized. This is Broca, who had found that there are areas here that are specialized for language. If you have a lesion here, then you have problems with understanding language, for instance. And today, the list of specialized areas has grown a lot. For instance, you have an area in your brain that is specialized for recognizing faces, just faces. This is Ramon and Cajal, who had studied the anatomy already, done beautiful staining studies. So they knew about neurons, dendritic trees, pyramidal cells, Purkinje cells, etc. This is Sherrington, who introduced the concept of synapse. Hodgkin and Axley, who studied modeled the tiny electrical signals that neurons use to communicate to each other. And on the theory side, you had a number of giants here, Gödel and Turing, etc. Shannon, the theory of communication was in place already, and von Neumann. And these gentlemen, McCulloch and Pitts, are the one who introduced the first model, simple model for a neuron, which is this, which is the building block of deep learning. Deep learning is just large networks of little computing units that look exactly like this, where you have a bunch of inputs, you have a bunch of weights on the connections, you take the average weighted sum of the inputs, and then you pass it through some nonlinear function. And there is maybe 5 to 10 nonlinear functions that are used today in deep learning, but basically think of this, for instance, as a threshold function. If the total sum is below some level, you produce a 0, otherwise you produce a 1, right? So very simple, a threshold gate. Electrical engineers would call this a threshold gate. So very, very simple computing units, and we stack them in complex networks to do all these complex computational tasks. But in 1950 already, you could start asking the question, where do you store information in your brain? Ask yourself, where do you store the first digit of your telephone number in your brain? Right? Well, people thought, and this is still the model we're using today, probably it is stored in synapses, because that's the place where you can rapidly influence neuronal activity, right? You can retrieve your telephone number in less than a second. So it cannot be stored entirely in the nucleus or in the DNA of neurons. Maybe there is a trace in there. That's a matter of debate. But definitely for influencing neuronal activity on a rapid time scale, the synapse is a good candidate, right? So that's the idea was born. Maybe we store information at synapses. That's fine, but how do we do it? Well, that's the question that we're going to deal with and come up with some possible solutions. But just to impress you that although this seems a very simple, like a threshold gate, right, a very simple computational unit, it's completely different and from other Boolean gates and from other ways of processing. It's completely different because you're storing information in these weights, in the synapses, and the storage occurs right at the same place where the computing takes place. So in a sense, it's very opaque. You have storage and computing that are completely intertwined, but on the other hand, it's also very efficient energetically. If you look at the beautiful supercomputers that you have here, or the IBM Watson computer, for instance, consume 80,000 watts, and your brain consumes between 20 and 40 watts. So 5 orders of magnitude more efficient, right? And one of the main reasons is that processing and storage are intertwined instead of being separated as you have in all the silicon models that we're using. So that's the first thing. The second thing is that the storage is completely different from what you have in your digital computer, where you have addresses and things are organized by addresses. Here, presumably, you're going to store your data. You're going to scatter the data across these weights and probably across many neurons. The first digit of your telephone number is probably distributed in many, many synapses in your brain, and maybe overlapping with other information about the second digit or other things. Right? So the storage, the mode of storage is completely different from what we have in silicon, and we need to think about that. All right, so how do we store information in these synapses? First person that came up with some vague idea was Hebb. He wrote a book. Hebb was a psychologist. There are no equations in the book. It's very verbose, but it was the idea, it's paraphrased in the little sentence that neurons that fire together wire together. This idea of correlation, that neurons have correlated activity, then the connection between them should strengthen. Okay? It's a natural idea, but it's very hard to see how, if you had, 10 to the 12 neurons all doing this little correlation algorithms, how can you have intelligence emerge from such a process, right? Nobody has been able to replicate or explain how this could be possible. So there's a lot to be explained about the idea of Hebbian learning. A few years later, Rosenblatt came up with the first algorithm for training a single neuron. It's essentially gradient descent. It's called the perceptron algorithm, but it's essentially gradient descent for training a single neuron. It was generalized by Widro and Hoff, but that's really a minor generalization. And then Minsky and Papert wrote this book in '69, Perceptrons, where they studied the properties of a single layers of such neurons, and they showed that they could do a lot of interesting things. But they also said, well, if you stack them, if you have a 2-layer, 3-layer networks, etc., we don't know how to set the value of the synapses, right? They couldn't come up with an algorithm, and they thought it was a challenge for the field. And this challenge was solved later in the late 70s, in the 80s by a number of people, but it's essentially doing gradient descent. Before I get there, let me mention that in the late 70s, Fukushima came up with this architecture, which is what we would call today a convolutional neural network for vision, where it's inspired by the work of Hubel and Wiesel in the 60s on the cat visual systems, where Hubel and Wiesel found out that cats had neurons that are dedicated for detecting particular features in the images, for instance, edge detectors, etc., in the low stages of their visual system. So based on that, Fukushima came up with this architecture where you have an input image, and then you have different layers of these convolutional filters. For instance, edge detectors at all possible location in the visual field, etc. And he said, well, you're going to have connections between these layers, and there is weights on those connections, and you are going to learn those connections using Hebbian learning. That's what he stated in his paper. Nobody has been able to do that, to do vision using Hebbian learning in this stacked architecture. And later I'll show you why. But it was a very, an idea that had a profound impact. Machine learning, as we know it today, started really in the 80s. There were a number of groups. This is some of the main group that had some significant influence. But basically, this group here is one of the main groups that introduced backpropagation, which is nothing else than gradient descent, stochastic gradient descent, which is the main algorithm for training these neural networks. So if you have a neural network like this, a feed-forward neural network for now, and you want to set the deep weights, this is the deep learning problem. How do you set the weights of the neurons that are connecting this layer to this layer, right? Well, if you have training data, you have examples of input and outputs of the functions that you're trying to learn. It could be vision, for instance, so you have images in the input, and in the output you have categories that you want to recognize in the images, cats, dogs, etc. You can set your weights randomly, you propagate forward, you get an error here. You take the derivative of the weights with respect to the error, and you change the weights in the direction that reduces the error. So basically, gradient descent done in a stochastic fashion, example by example, rather than in a batch fashion. But that's the basic idea of backpropagation. This is the algorithm that is used throughout deep learning, right? So very simple idea. Newton and Leibniz could have found the equations for backpropagation, right? The equations for backpropagation look like this. So the adjustment of this weight is equal to a learning rate that you can adjust time a postsynaptic backpropagated error and a presynaptic activity. So this weight here, to change this weight, you just have to change it by a small amount, which is the product of the activity of the presynaptic neuron. So this sounds like a Hebb, times the backpropagated error, which is something that comes from the top layer where you compute the error and then you send it back through the layers. Send it back means that you are multiplying it by the transpose of the forward matrices, basically. So you're sending it back through the same network, and every time you traverse a layer, you have to multiply by the derivatives of the transfer functions. But basically, it's just an application of the chain rule. And again, this is what is used today in these computer vision systems that are outperforming humans at recognizing categories. And you use these convolutional architectures that are very much like those that Fukushima had introduced in the late 70s. Okay, so let me say a few words about theory. Some people complain that we don't know anything about deep learning. It's all black box engineering. That is not true. There is actually quite a bit of theory. I don't have time to review it entirely, but I'll just show you two little pieces of theory. The second one will be important in the applications I'm going to show you. So the first little piece I'm going to show you is what I call learning in the machine, which is a way of looking at neural network where you really take into account the physical constraints of the world in order to better understand the constraints on learning in biological or in neural networks. And you will see that this is going to explain why Fukushima the idea of training weights in a Hebbian fashion did not work, and will give you a sense of why there are so few learning rules, right? Basically, I showed you two rules, gradient descent and Hebbian learning. Why haven't we found other learning rules? Is gradient descent the best you can do, for instance, right? Etc. So I'm going to answer those questions for you. So, Einstein, when he was asked, how did you find out about special relativity? He said, well, my technique was to think I was a photon or think I'm a ray of light, right? So it's imagination, putting yourself in the shoes of the objects that interest you. So let's do the same thing. Let's think you are a synapse, okay? Think you are a synapse. And a synapse is a very small object. So probably not used to think about objects of that scale. So we're going to multiply by a factor of 10 to the 6, 1 million. If you do that, a synapse becomes the size of my fist. Okay? So here's a synapse in your brain, and you are trying to learn how to play tennis or how to play the violin. Okay? Where is the tennis racket? Any guesses? No, not that far. 10 to the 6, the tennis racket is in New York. Okay? And now this synapse, its task is to strengthen itself or weaken itself so that you play better tennis. This synapse doesn't know anything about tennis. It doesn't see the outside world, right? The only thing it sees is protein, RNA, etc. in its small environment. Okay? How is it going to know whether it should strengthen itself or weaken itself? Okay. That's the deep learning problem, right? How deep parameters of a model, very far away from input and outputs, can make a decision on what to do in a completely blind fashion. All right, so if you start thinking in this way, you realize that in a physical system, a learning rule has to be local. It can only depend on local variables, right? So in these very simple, simplified neurons, you can say that a learning rule for adjusting the weights, of the weight of a synapse between neuron I and neuron J, should be a function of local variables, and you can decide what you call local variables, but a reasonable choice would be the activity of the pre- and postsynaptic neurons and maybe the weight itself, right? Now, if you are at the output layer, and again, I'm just thinking about forward networks for now, you may also have targets, right? You may know what is the right response at the output layer. Of course, you don't have this for the deep layers, right? But you still could imagine having local rules at the top of the network where you're allowed to include a target among your local variables. Okay? So this very simple idea is actually very useful because now you can stratify learning rules. I can say, well, I'm interested in learning rules that are polynomial and maybe low-degree polynomials, up to 5 or 6, right? So now you have a space of learning rules and you can study them using differential equations. You can study them on linear units, in some cases in non-linear units, and look at the effect of local learning rules. And we have done that for polynomial learning rules up to a certain degree. And you can see that there are learning rules that allow you to extract interesting things, such as the center of gravity of your data, or maybe the principal component of your data, etc. So you can extract, using these local learning rules, interesting statistical properties of your data. However, you are never going to be able to learn complex function using simple local learning rules. So what I'm saying to you is that if you have a feed-forward network like this, and you have local learning rules between the layers, where in this case, for instance, this learning rule depends only on the local variables, then, and no matter what the local learning rule is, no matter if you use polynomial of degree 125, it doesn't matter. This system, this learning system, will never be able to learn complex functions. As a special case of this, Fukushima, who wanted to do computer vision using a convolutional architecture and Hebbian learning, which is a special case of local learning, that program cannot succeed. And the reason is simple, is that if you write down the equation for the optimal weights, that is, the weights where the derivative of the error with respect to the weights is equal to 0, you write down those equations, which is basically backpropagation equals 0, you see immediately that the deep weights have to depend on various things, including the targets. Every deep weight in the network has to depend also on the targets. Okay? If you do local learning stacked up in the way I was describing to you, it's impossible for the deep weights to depend on the targets. So in order to have optimal learning, you need to have a communication channel that sends information all the way from the target down back to the deep synapses. There is no other way. That's what we call the deep learning channel. And of course, once you have this inside, you can ask all kinds of questions about deep learning channels. So where is the channel physically? In a physical system, where is it implemented? In biology, we know that we have all kinds of feedback pathways. So maybe some of them are carrying the information that is needed for deep learning. Then you can ask what kind of information is sent over these deep learning channels, et cetera, et cetera. And so we have been studying this. If you're interested, you can look at this paper. But one of the results that come out of these analyses is that backpropagation is pretty much optimal. So it is unlikely that there are algorithms that are much, much better than backpropagation for training deep neural networks. Okay, second legal piece of theory, it's how do you design recursive networks? So now I'm moving to networks that have, recursivity. This is important because there are, for many problems, you have data that has a fixed size, right? Like images, or you have, sensors. You have 100 sensors, and you collect data at different times from these 100 sensors. All your data is going to be in the form of vectors of dimension 100. However, there are other problems where the data does not have fixed size. It varies in size. In addition, it has some kind of graphical structures. For instance, molecules. If you look at small molecules, little graphs, they vary in size, right? You're doing bioinformatics. You have DNA sequences, protein sequences. Sequences have different length, and they have a structure, sequential structure. Of course, the same thing in language. Or you can look at parse trees. Then you have little trees, et cetera, right? So lots of situations where your data doesn't come to you as vectors or tensors of fixed size. In those cases, instead of using feed-forward networks, you want to use recurrent networks. How do you build them? I'm going to show you that there is essentially two techniques you can use to build interesting recursive architectures that I call the inner and the outer approach. So in the inner approach, what you do is you use neural networks to crawl the graph of the data. You are inside the data graph, and you crawl the graph using the neural networks. Of course, I'll give you examples in a second. In the outer approach, you're using neural networks in a direction that is perpendicular to the data, to the graphs associated with the data. So for instance, on a sequence problem, how does this translate? For instance, if you have a sequence, typical model for a sequence, a generative model would be an HMM, right? Or input-output HMM, etc. So an HMM is a probabilistic model where you have hidden states here. You have transition between the hidden states, and every time you are in a hidden state, you're producing a symbol of your alphabet, right? So that's called the Bayesian networks or a graphical model. You need to have a directed acyclic graph, such as this one. And if you want to refactor it using neural networks, what you do is you put neural networks on the edges of this graph. So you're going to have a neural network here for the transitions. And if you reuse the same neural network at different locations in time, that's what gives you the recursion, right? Because the output of the network goes back into the input of the same network. So this network and this network have the same weights. And then you have a network here for the producing the letters of the alphabet. And the same weights are used in these different neural networks in general. And so this entire HMM can be rewritten in the form of two recursive neural networks. If you want to do an outer approach, so you see in this case the neural networks are embedded essentially inside the graph. They are crawling the graph inside the edges of the graph of your HMM. In the outer approach, you have your sequence here, and you're growing your neural networks in a direction that is perpendicular to the sequence. So you have a first layer of neural networks, which maybe are looking at a symbol and its two neighbors, and then the next network looks at this symbol and its neighbors, etc., right? And produces a vector here, an output vector. And then you have a second stage of green networks, where the green networks are looking at several outputs from the first layer. So the output of the green network will depend on a window of inputs that is larger than the blue networks, etc. Right? So as you go up in this stack, the networks at the top can see longer and longer portion of the input sequence, right? And you can do weight sharing. They can have the same weights within one level of the stack, or you can also relax that assumption if you want. But you can stack those and again get outputs at the very top and backpropagate gradients through the entire stack if you want to train these networks. So I'm going to show you in the applications examples of the inner or outer approach applied, for instance, to molecules or proteins, etc. Okay, so let's move to application in physics. There are many, many applications of deep learning in physics. It's just the beginning. You can work at the very small scale, particle, subparticle scales, at the very large scale, cosmological scale, and everywhere in between. For instance, at this scale, you have images, so it is natural to use, to try to use convolutional neural networks applied to these images to, for instance, detect quasars, galaxies, lensing, etc. Or you can work at the examples I'm going to show you are more at the particle scale. We work with the Large Hadron Collider, with ATLAS at CERN. But anywhere in this very large range, there are interesting problems where deep learning can be applied. Now, at the small range, we have worked on actually different problems. Most of the work has been done in high-energy physics, but I've also been working on problems in antimatter physics, detecting anti-hydrogen, or in neutrino physics. And from a machine learning perspective, all these are pretty much the same. Because what you have beautiful detectors that have complex geometry, and the geometry may vary from one experiment to the other, but you have these detectors. And these detectors basically produce images, right? Energy deposits, etc. So you get images, and if you want to work at the image level, you can start here. And you can use convolutional networks, or you can use other types of networks. The questions you want to ask there is, first of all, does the geometry of the detector changes things, right? If it's not planar, if you have detectors on a sphere, for instance, do you have to worry about the curvature of the sphere? Does it have any effects? And you want to worry about invariances. If your recognition, if the problems you're trying to detect is translation invariant, then it makes a lot of sense to use convolutional neural networks. If you don't have translation invariance, then maybe you want to relax that assumption. So those are principles or ideas that can guide you in designing networks to process things at the level of images. The next level, physicists have developed all kinds of algorithms for processing these images and detecting interesting substructures within these images, things like jets, things like tracks, things like vertices, right? In fact, the software that we're using for in antimatter data to detect these kinds of objects is stolen from the software using high-energy physics to detect vertices and tracks, etc. So you can reuse the software across these different experiments. So if you work at this level, you're going to get a list of objects, and the list has variable length, which tells you you're going to have to use recurrent networks, right? So you're going to get a variable list of, let's say, vertices and tracks. And then you have primary vertices and primary tracks and secondary vertices, etc. They may be described by different sets of variables. So they may also have different lengths. So one more reason for using recursive networks if you work at this level. And then from this list, physicists very often can derive vectors of fixed length, representing, for instance, the momenta of the particles. And then you can use, again, feed-forward neural networks to process these vectors of fixed length and produce predictions or classification, regression, etc., depending on the problem you're interested in. So we have done all these, or started doing all these on these different experiments. In the interest of time, I'm just going to show you the result of one experiment done at this level here. So this is on high energy physics with Atlas. If you're interested in the other types of representation, etc., you can look at some of our papers here. But basically, in this example, the data comes to us in the form of millions of examples, which can be produced because there are good simulators for these collisions. The vectors have typical length 20 to 30, again representing momenta of particles. And we see that deep learning in general outperforms the methods that are currently used by physicists, things like decision tree, for instance. So the first example we started with was detecting Higgs bosons, for instance. These are the software that are that were used to produce the data. For those of you who are, experts in high energy physics, I know that there are different simulators. Some have better accuracy than others, and they are slower, etc. So these are not the most accurate simulators, but they are fast. So we could produce a very large number of examples for training a classifier that detects. Separates Higgs bosons from events that do not contain Higgs bosons. The work was done with Peter Sadowski, a previous graduate student of mine, and Daniel Whiteson, who is a physicist at UCI from the Atlas consortiums. So in this particular case, 11 million examples, inputs are vectors of length 28, including 21 low-level features and 7 high-level features derived by physicists. You can see some of the histograms of the distribution of these different features. But the bottom line, these are details of the neural network we use, the kinds of things that people like to hide because they're not so interesting, and you cannot really justify everything other than you tried a lot of things and this is what worked. But basically we tried a number of layers, up to 6 layers, different types of pre-training, weight decay, dropout, etc. I won't go into all those details unless you're interested in. But the bottom line is an 8% improvement in the area under the curve, under the ROC curves that you see drawn here. So this is a typical curve for a classifiers. You can see different methods that are here. And the better methods are those that have the largest area under the curve. So you can see deep learning is here. And compared to this other method, including shallow networks and decision networks, etc. So this is a significant improvement. And we have shown similar things for other exotic particles and other types of experiments. You can also see, at least in this case, that if we train the network without the high-level features derived by physicists, we can get almost the same performance. Not exactly the same, but very close to the performance that you get by including the high-level features derived by physicists. Let me quickly shift to chemistry. So one typical problem there, you have small molecules, you want to predict their properties, right? Physical properties, chemical properties, biological properties. It's almost endless, both in terms of the space of a small molecule, which is very large, something like 10 to the 60, and in terms of the features. In principle, you could look at many different features. The main problem there are the training sets. There are not many data sets, large data sets of molecules annotated for some chemical properties, whether it's solubility or melting point or whatever. The data sets that are publicly available are usually small, but we can still work with them. The interesting thing is that small molecules can be represented in many different ways. This is the usual molecular graph, if you want, representation. But of course, you can represent them as 3D objects. So you could have the XYZ coordinates of the nuclei of the atoms of these molecules. Note that this is going to be a variable length list, because molecules could have different numbers of atoms. You can represent them using so-called smile strings. So this is a smile string representing this molecule. It's very easy to learn how to speak smiles. You have this nitrogen is here, goes here, it can branch left or right, so you open the parenthesis. This is a carbon. If you don't see anything at the vertex, it means it's a carbon. Then you go in this direction, you have another carbon. You go here, it's an oxygen. It's the end of the branch, so you close the parenthesis. You don't count the hydrogens. You go back here, now I go here, it's a carbon. Now I can branch again, so I open the parenthesis. You have a double bond, it's the equal sign. You get the oxygen, it's the end of that branch. You close it, and then you go here, and it's the last oxygen, right? So very easy to see how to build these strings. So if you like to work with strings, you can work with small strings. Of course, they are going to be of variable length, depending on the molecule. And then, of course, the last representation is fingerprints, where you have vectors of fixed length, usually very, very sparse. Depending on which basis you use, these vectors can be very long, up to 1 million, for instance. And the 1 means that a particular feature is present in the molecule. So as a feature, people today typically use combinatorial features. So you can look at all path of length 10, labeled path of length 10, carbon, carbon, nitrogen, carbon, etc., right? You take this combinatorial basis, may have a very large size, as I said, up to 1 million, and you index your molecule in this way. Obviously, this is fixed size, so you could use a forward neural network on the fingerprints, but it's not a good idea because these vectors are also very, very sparse. So you probably want to do first a compression step using a random projection or a compressive autoencoder, for those of you who know these things, to first compress these high-dimensional vectors into lower dimensional vectors, and then apply a feed-forward network to the low-dimensional vectors. Well, as an example, let me show you what you can do if you want to work with the graph. I told you about the inner and outer approach. The inner approach requires a directed acyclic graph, and this is not a directed acyclic graph for 2 reasons. The edges are not directed. There is no obvious orientation for the edges in a molecule. And you can have cycles, right? You can have benzene rings, etc. So to deal with this, what we do is the following. We take the molecule and we replicate the molecule as many times as it has the number of atoms in the molecule. In each replica, we choose a different vertex as the sink, and we orient all the edges of the molecule towards the sink. So you have a gray sink here, all the edges are oriented toward the sink. This is what gives you an acyclic orientation, and you do this in all possible ways. And now you can place neural networks inside these graphs. So here, for instance, you have a neural network that is going to crawl starting from this hydrogen and produce a hidden vector here for these carbons, and then repeat that in all positions of this molecule. The same thing occurs here. At the end of this crawling process, you are getting a vector in the output of each one of these crawling processes. The vectors are aggregated here, and these are vectors of fixed length, which you then can pass in a final feed-forward neural network, which produces your prediction. In this case, it was solubility, but it can be anything. And because everything is acyclic, you can backpropagate the gradient through this entire architecture. So that's the inner approach for small molecules. If you wanted to do an outer approach, you would have the molecule in the plane, and then your neural networks are growing in a direction perpendicular to the plane. And in the first layer, you have a neural network that, for instance, is capable of looking at one atom and its immediate neighbors at distance 1 in the graph, etc., right? And should you use the inner or outer approach, there is no general, sense that one approach is better than the other. It really depends on your problem, on the software libraries, how easy it is to encode a particular approach or another one, etc. And we are going to release on GitHub software that does both the inner and outer approach for small molecules, and will be available to the community to use. But again, the main problem is the data. It's not the machine learning. Other very important problem in chemistry, predicting chemical reactions. You can approach this problem from different angles, QM, rule-based system, deep learning, etc. We tried all of them. QM, at least in our hand, doesn't work, especially if you want to do things in high throughput fashion and fairly large molecules, etc. So we wrote, my student Jonathan Chen wrote a system of rules, about 1,800 rules that cover the sort of undergraduate curriculum in organic chemistry. Basically, you can write reactions using a slight generalization of SMILE strings, where you have SMILE strings for the reactant gives the product. Here are the SMILE strings for the product. The key difference is you have to number all the atoms so that you have an atom mapping, proper atom mapping between all the atoms on the left-hand side and all the atoms on the right-hand side of the molecule. So we built this system, and it's actually used in education. It's distributed by Wiley because we turned it into an interactive system where students can practice how to predict chemical reactions and how to do it in the reverse direction for retrosynthesis, etc. This is nice, but the problem is that the system is not very scalable because it's very tedious to add these rules by hand. Every time you add a rule, there is a chance that it may break a previous rule, so you have to be very careful. And it's not very scalable, and it covers only undergraduate chemistry. Of course, we would like to reach human-level chemistry and even beyond. So what we're doing currently is using deep learning in the following way. How does a chemist think about a chemical reaction? Well, it's basically electrons moving around, right? Nuclei are unchanged. You have electrons that are moving from sources to sinks, and maybe in a reactions you may have several such movements. So this is very simple reaction. Is the bromination of an alkene. An alkene is a molecule, is a small molecule with a double bond. So you can see here that first you have HBr gives you H plus Br minus, and then the Br minus, the bromine attacks this carbon and produces the brominated compound. So basically what we do now is we have reactants. We look for sources and sinks of electrons. This is no machine learning. You just have rules for finding potential sources and sinks. So let's say I find 10 sources and 10 sinks. Now I have 100 possible source-sink combinations. I have to rank them. And ranking is a machine learning problem. It's what Google does when you enter a query. You get a rank list of answers. So we use deep learning to do the ranking. And the way it's done is using what is called the Siamese network, where you have a deep network here, but these two branches at this level are identical, and we put a source-sink combination on the left, a source-sink combination on the right, and the output has to tell you whether the left combination is more favorable than the right or not. And so we train this system, and it's doing better than the previous system. It's reaching roughly the level of a graduate student in organic chemistry. It's not yet at the level of professors, but that's where we're going with this. And again, the main problem is the data. Give me training data for this system, right? There is no publicly available database of chemical reactions, period. It's a very, very unfortunate situation. All right, let me finish with biology. There are many applications of deep learning in biology. You can work at the DNA level. You can try to predict the effect of mutations. You can try to parse genomes or promoters and identify the location of transcription factor binding sites and all kinds of interesting things. I'm going to show you applications to protein structure prediction. This is a big center here for solving protein structures experimentally, but I'm going to show you methods for predicting structures using deep learning. So the key problem with protein structure prediction is that the structure is invariant under translation and rotation. So if you took the sequence and tried to predict XYZ coordinates directly from the sequence, it would be almost impossible for a neural network to do that, because if you move the protein around, the structure is the same, the XYZ coordinates are completely different, right? So to factor out that big problem, we break the prediction into 3 steps. We start from the sequence. We predict a number of features along the sequence, such as secondary structure, you know, whether a given amino acid is an alpha helix, a beta strand, or a coil. You can predict relative solvent accessibility, whether a given amino acid is on the surface of the protein or in the core, etc. So that's what you get in the first stage. In the second stage, we predict the contact map, which is basically a translation, rotation, invariant representation of the structure. So If you're not familiar with contact map, basically imagine having the protein along the x-axis and along the y-axis, and the contact map, it's this very sparse binary symmetric matrix where you put a 1 in position ij if and only if amino acid I and amino acid J are close to each other in 3D. Technically, it means the carbon alphas of the two amino acids are within, let's say, 8 angstrom or 10 angstrom, depending on the cutoff you want to use, right? Or the carbon betas, same thing. You can have also a coarse contact map if you look at the secondary structure elements, so whether two alpha helices are close to each other or not, and et cetera, et cetera. And then from the contact map, you want to retrieve the 3D structure of the backbone, possibly also of all the side chains, et cetera, right? So the good news is that the third step is essentially solved. If you have a good contact map, you can retrieve, let's say, the coordinates of the backbone up to mirror images. This is not machine learning, it's just techniques from distant geometries. It's what you do when you do NMR, etc., right? So the third stage is solved. Let's look at the first stage, which is essentially predicting secondary structure. You can view it as a translation problem, going from a language which has 20 letters, the 20 amino acids, to a language that has 3 letters, alpha, beta, or coil. So it's a translation problem. Again, you can do an inner approach using, in this case, what is called a bidirectional input-output HMMs. HMM, for those of you who like graphical models, but basically I'm going to rewrite this system using 3 neural networks. One neural network that predicts the output as a function of these 2 hidden vectors and the input, which can be a window of amino acids, for instance. And then the backward chain, you have a hidden vector here, which is computed by a neural network as a function of the hidden vector in position bt plus 1 and the input, and similarly for the forward chain. So 3 neural networks. You unfold them in time, you get this. Again, everything is acyclic. You have the output here where the targets are known. You can backpropagate the information through this structure. I won't go into the details, but to give you a sense of how performance has improved over the last 40 years, Chu and Fassman built the first system for secondary structure prediction in '78 with an accuracy of about 60%. Kian and Sinowski in '88 trained the first neural network, not a very deep one, on prediction, and they got about 64% correct prediction. And our latest system is about 95% correct using these recurrent deep learning approaches. And we don't think you can improve this by much for a variety of reasons. You know, some proteins do not fold spontaneously. Some proteins need chaperones to fold properly, or they need ions, particular ions or metal atoms in their environment, etc. So, and all that information is not in the data. So we think the problem is essentially solved. And yes, deep learning contributed to this progress, but most of it comes from the data, actually. It's the fact that the PDB has grown a lot, and by several orders of magnitude, and so we have much bigger training sets, and that's what really help us improve the prediction. So first problem is solved, third problem is solved. What about the second problem, prediction of contact map? That's the one that is still unsolved, and it's the hard problem in this pipeline, of course. The way we approach it is, again, using an inner approach, which in 2 dimensions is going to look like this. You're going to have an output, which is the prediction of whether I and J are in contact or not. You have 4 hidden vectors in these 4 hidden lattices, where you propagate information laterally along the 4 cardinal points, and then you have your input here. So this is the system. It reduces to 5 recursive neural networks, and that's what you get when you do this inner approach. I'm skipping all the details. If you want to do an outer approach on contact map prediction, we have done that too. Imagine having your contact map in the horizontal plane. You're going to build neural networks on top, stack them on top. Imagine the protein as it folds in time. You get a slightly different contact map at each folding step if you want. You don't have targets for all the intermediate steps in general. You don't have movies of proteins that are folding. You could try to produce those with molecular dynamics or something like that, but you certainly have targets at the very top where you have the folded structure, and you use that to backpropagate the gradients throughout the stack, and so we have developed this approach. A CASP experiment a few years ago where people compare different predictors for difficult contact maps. You can see that the deep learning methods are the top, which is a good news, but the performance, the accuracy is about 20% on the difficult contacts, the one that are, long-range contacts, amino acids that are far away on the sequence but close to each other in 3D. And so that's where improvement needs to be made to bring that number to about 50%. And again, data would be crucial in achieving this. It's not so much the machine learning methods. I'm going to skip applications to circadian rhythms, which is something we're working on. Let me close by a couple of examples of applications of deep learning to medical images. I think that's a very easy area where a lot of progress can be made rapidly. The big problem, again, is aggregating data across hospitals and convincing the medical community to use these methods. But just as an example, here we're looking at mammograms, X-rays of breast to detect breast cancer. Here, but we're not detecting breast cancers. We're looking at the vascularization of the breast and detecting calcium. So if you have a high level of calcium, it's a potential sign of heart disease, right? Calcification of blood vessels. So we have trained this network to measure the overall level of calcification that is seen in these images. So it's a regression problem. We have a small data set of annotated data where radiologists have done all these estimates, and we get very good predictions using standard deep learning methods, standard convolutional neural networks. Another example is a system being developed in the lab of Chris Hughes on my campus, where he has a microfluidic system where he can grow blood vessels in these little chambers, and then he can inject cancer cells and drugs and see how these various elements interact. You can see the cancer cells here in blue, so you can inject a drug, and then you can ask, did the drug affect the cancer cell, did it affect the vascularization, etc. This can be done in high throughput fashion. You can produce thousands of such images per hour. It's very tedious for humans to look at them and decide whether, the cancer, the number of cancer cells has been reduced, etc., etc. So again, using deep learning, it's fairly easy to develop very accurate methods for doing that once you have a labeled training set. So we have undergraduates that are labeling the data so that we can train the systems. This is a system for detecting metastasis in the spine. Most cancers tend to have to metastasize to the spine, so you would like to automatically detect on these images of the spine if there is a cancer lesion and where it is, etc. Again, deep convolutional networks work very well. And the last example I'll show you is polyp detection. We have a small data set of 6,000 images of polyps, 6,000 images without a polyp. We can use deep learning to both recognize polyps and find bounding boxes. There is a technique that allows you to find bounding boxes around polyps, where at the next stage, we're trying to get also annotation of polyps that are benign versus polyps that are malignant to refine our classification. Now, you may say, that's very nice, but when you do a colonoscopy, it's video. You're doing images. Well, the key point is that training neural networks may take days, but once they are trained, they are very fast. So on one image, when I feed into a trained neural network, I get the output, the prediction, within 20 milliseconds. So I can easily process 10, 20 frames per second using this system trained on image, and that works very well, actually, also on video. And so that's a system that we're currently developing. Just to give you an idea, the area of your colon is the size of a tennis court, and a colonoscopy is done just in a few minutes, right? So you can imagine that physicians occasionally can miss some corners, and so having such a system that beeps or potentially alert a physician to the potential presence of a polyp and maybe even classify the polyp into malignant or non-malignant could be quite useful. Thank you very much, and I can try to answer any questions. So if you want to open the black box, you can do it. You have to do neurophysiology on your neural network, which means you have to go neuron by neuron, perturb the inputs, and see how the neuron respond. It is extremely tedious, extremely time-consuming, and very few people can do that. In some cases, in convolutional neural networks, just for the first layers, the first layers where you have these convolutions, you can visually, plot the receptive fields of the neurons and inspect them by eye. So you can do a little bit of inspection, but there is nothing that on a large scale is going to tell you what is the neural network doing and how it is doing it. And I think this is should not be actually too surprising, because it's part of this opacity of the fact that when you train a neural network, you are storing your training set in the synapses of the neural network, and this is a very opaque process. Each training example leaves a little trace through the gradient descent procedure, leaves a little trace of itself in each one of the weights of the network. So each network knows a little bit about each training example, and that has to be very, hard to invert. And finally, every day you use your brain and you trust your brain, you have no idea how it really works. So you should not be too, obsessive about understanding neural net. And I don't know how my car or my computer works, to be honest. So, but yes, ideally you would like to understand everything as a physicist.
The whole basis of science is to be able to make testable predictions, not simply to be able to make predictions.
No, the predictions are testable. The predictions are testable. I mean, this is how you do, validation, cross-validation, etc. You need, of course, ground truth from expert or from some other sources, but you can test the quality of the predictions of a neural network.
We have a fundamental epistemological issue here in that a human being can look at data and deduce the standard model. A neural network looks at data and it deduces what?
It is possible down the road to have a neural network that looks at data and deduces the standard model, but we're not there yet. But things are moving in that direction.
I think this ties off the previous question of how do you go from learning? I can teach you a set of rules, let's say for a game. Oops, sorry. I can teach you the rules of a game, like let's say chess. But I can't. How do you then go from learning the rules and maybe even an optimized algorithm of doing that to thinking outside of the rules that I gave you to find a better way to get to the solution? Like, is there progress towards that? Like, I mean, obviously, you get to the end game of will machines eventually overtake us and kill us all because they figure out that we're useless. To get there, they have to think outside of the inputs that we're giving them. So is there any projection or thinking beyond the confinement of the boundary of the problem that
you're doing? Well, first of all, when you speak about chess, there is two things. There is the rules of the game, and then there is the rules or policies that allow you to win the game, right? And machine learning is very good at that. Chess is essentially solved. And the same thing for Go. Actual human, it's not clear what, Go experts, it's not clear what kind of system they use to play Go. And just by looking at examples, we have systems now that play Go better than human players, which is a very difficult problem. So yes, I think that deep learning can over time become creative or able of going outside the limited data that it has used for training, as long as it reuses other data sets, as long as it can transfer learning inferences, make analogies, etc., which is what our brain does, right? So it is possible, and it, you know, we see the beginning of that in different areas. Weight decay is a regularization, mathematically it's a regularization procedure. So imagine having a prior on the weights that is a Gaussian prior to constrain the weights. Then if you write the equations of gradient descent, you're getting your learning rule minus alpha times the weights, right? The derivative of W squared. When you take the logarithm of your Gaussian, you get a quadratic form W squared, you take the derivative. So essentially, that's the weight decay. It prevents the weights from becoming very large or very small. So it's a method of regularization. So that's weight decay. Oh, that's a very nice question. Yes, there is some work, but much more could be done. So a beautiful problem is, that I thought of, that I even worked on, but one could try, is you take Mathematica and you generate lots of examples of symbolic integration. And then you try to learn the rules for symbolic integration using deep learning and recursive networks. But that's just an example of something that someone could do. Yes. So we've done that. So you're talking about connecting the output layer directly through the deep layers, keeping all the intermediate layers, and it's essentially equivalent. Yes.
As a classifier algorithm, what about uncertainty? Because the issue in that particular topic is not just that you get an 8% improvement in characterization as signal, but also you have to understand the uncertainty with which you've done that. Are there theorems that can allow you to control those systematics?
Control them, it's a difficult question, but you can certainly get a sense of what is the uncertainty on your prediction by having, you know, a all-out data set on which you know what the answers are, and you have the labels, and you test the performance on that data set, and there you get a sense of the level of uncertainty in the system.
So it basically goes back to the uncertainty associated with your training data set?
Yes.
Okay.
So any modeling is part art, part science. And how much the result of a machine learning study depends on the expertise and intuition of the user. For example, you mentioned an optimal number of hidden layers. How do you select that? What happens if you deviate from that?
Right. So, you know, there is a number of general principles that have been emerging, or general techniques. For instance, for images, it's very natural to use convolutional neural networks. I showed you the inner and outer approach as some kind of guideline for building recursive system that can deal with data of variable size. So there are all kinds of, you know, principles and rules, but there is nothing that tells you this is the best architecture, right? And a lot of it requires some tinkering or trial and error or so-called hyperparameter optimization. And you know, there is no common solution, no agreed solution on the best way to do that. There is some, you know, some hand and crafting involved, and it's a little bit inevitable, I think. And yes, so experience with training this system is certainly a helpful skill. On the other hand, there is all kinds of software and libraries and tools that make the task much easier today than it was, say, 20 years ago. So if you use TensorFlow, you use things like Keras, those are tools that allow you very rapidly to adopt and deploy these techniques.
When you think of a neural network, we should create other successful neural networks.
Yes, I can think of that.
Hi. Does anyone, say, take a neural network and model the brain and try to correlate the number of layers in the neural network with something like types of synapses or something?
Absolutely.
Like that?
There is a whole literature on that. In particular, in the visual system, people have shown that if you build a visual system, let's say, using these artificial neurons, but which has the same number of layers roughly as you have in the biological case, etc., you end up observing in the artificial system a lot of the characteristics that are in the biological system. You find the same receptive fields, etc., etc. So a lot of that has been done, yes.
In the brain, what are the mechanisms that it uses to do the feedback?
Well, we know that there is a lot of feedback connections in the brain, right? That is well documented. But we don't know, for instance, if which feedback connections are carrying an arrow signals versus other types of information, because there's the word feedback is very ambiguous, for instance.
But you're saying basically there are, well, no, let you keep going.
There is a physical pathway, a series of axons that goes essentially from the output level, from the motor level, or from the input level, all the way down to a synapse. There's actually many such pathways. Which one are used for learning, that we don't know. That is still things that neuroscience has to sort out. But the channels are there, that's for sure.